So today we're going to look at your revision paper set 1 booklet B. Okay, now the first question is actually a very simple question. You should have read this question already. So in terms of circulatory system, they are testing you on whether do you know the directional flow. So let's talk about that first. While I'm talking about it, I will also annotate what are some of the key ideas so that you can relate to the blood flow. Now, so this is actually your heart. To identify the heart, just know that blood actually flows through your heart twice. Okay, I'm going to show you where is this twice. Let me zoom in. Now, let's talk about blood flow first and then we do our annotations. So let's just say we start off from the heart. Now your heart, when it first starts off, I'm going to use red to denote oxygenated blood. It will flow up to your heart to your small intestine. Now your small intestines can also be counted as any other parts of the body. Alright, that's fine. Now inside, a very important process actually takes place. This is known as your respiration. Now I've told you many times when it comes to respiration, it is very important to write down the equation to help you. So the equation for respiration would be that, okay, it will take in glucose, which is your digested food as well, together with oxygen, and then it will give out carbon dioxide together with energy and water. But the key thing that you always want to remember would be your energy and your carbon dioxide. Now your energy is what you need. Okay, this is very important. Please pen this equation down. It is so important because it's able to explain to you what, why, or account for why the difference in concentration of your oxygen. So please annotate everything that I have written here. Now, for your carbon dioxide, this is your waste product or one of your waste product that needs to be removed. So once you know this equation, you will also know that your small intestine will carry out respiration. So in the process of respiration, it will give out carbon dioxide and you will use up all your oxygen. So remember I told you initially, this is actually oxygenated blood going this way. However, once it comes back from the other parts of the body, it becomes deoxygenated blood. Why? Because your small intestine or other parts of your body would have used up your oxygen for respiration. In exchange, it will have given a lot of carbon dioxide into the blood. So blood that is flowing here, we can say this is oxygen rich and carbon dioxide poor. But once it passes this point here, okay, the opposite is true. This is now carbon dioxide rich and oxygen poor. Now, because it's now poor in oxygen, it will return to your heart. Now, at your heart, right, your heart will actually pump the blood back to your lungs, where it carries out this very important process known as gaseous exchange. Now, during gaseous exchange, what is removed would be your carbon dioxide, and what is taken into the blood would be your oxygen. Now, it is very important to mention in your answers that the oxygen goes into the blood. If not, there is no point. You cannot just simply say, oh, oxygen goes into the lungs, full stop, or goes to the heart. No, it needs to go into your bloodstream first. And then it gets pumped from your lungs, okay, to your heart. So let's recap. So if I look at the flow of heart from your heart to all parts of the body and then back to your heart, this is the first time. The second time would be from your heart to your lungs and then back to your heart. So this is the reason why we say it's a double circulation. 2 times. Okay? So the question asks, substance X is found in the blood. Blood at B contains higher amounts of substance X than A. So like just now I have explained to you, what is B rich in? B is rich in carbon dioxide, whereas A is poor in carbon dioxide. So substance X has to be carbon dioxide. Please spell out the gas in full. You're not allowed to use the chemical symbols. Next, compare the amount of oxygen. Now be very careful because the question specifically asks for oxygen. If you compare anything else, digested food, carbon dioxide, everything else is wrong. Okay? So take a look at here. So C would be your deoxygenated blood, and D will be your reoxygenated blood or oxygenated blood. Okay, so therefore D has a higher amount of oxygen than C, or C has a lesser amount of oxygen than D. Okay, so I would say blood. Okay, I would just say C or D has a higher amount of oxygen than C. That's about it. Okay, annotate everything that I have written down. Next. Okay, this is a very, very common question. You must know how to do it. Now, most students can get this right. The only issue would be that you are very, very careless. Alright, I will tell you where is your carelessness. Now, they tell you that this graph below shows John's heart rate from 1 p.m. to 2:30. So he said he did 3. So right off the top, you would know that the one that actually has the highest amount of, or rather the highest rate of heart rate, that would actually be your running. Okay, and you need to explain it to me why later. So what was John doing from 2 to 2:30? So obviously he was running. Right? Now, why did John's heart rate increase during the time period from 2 p.m. to 2:30? Now, this has got to do with the fact that your body needs to carry out more respiration in order to meet the energy demands. Right? Okay, now, so how come his heart rate increased? The first thing you need to say would be that, maybe I break it up into 1 point by 1 point. The first thing you need to say would be your heart, okay, or his heart pump faster. This is the first key point. Now, please don't say his heart beat faster. His heart beat faster, not really encouraged. Use the term pump faster. This is your first key point. Your second key point would be to transport oxygen and, or rather, sorry, can I undo this? To transport more oxygen and digested food. By the way, DF stands for digested food. These two things are extremely key in your answer. If you are missing any one of these, oxygen or digested food, you don't get the marking point for this. You get a deduction. Together, it is 1 point. And then after that, okay, to where? To muscle cells. Now, if you say to all parts of the body, we will accept it. However, I want to train you to be discerning, to be able to understand that it is the muscle cells that needs more energy because you're talking about an exercise, right? Okay. Now, how come you want to transport more oxygen and digested food to muscle cells? This would be to hence increasing the rate of respiration. Very important. I must see that there is this keyword increasing and rate of respiration, because whether you are running or you are not running, you basically would still need to respire. Doesn't mean that you're sitting down means that you don't need energy, right? That's not true. So because of that, you have to say the word increase, right? That is the difference. It's not have and don't have. It's one is faster than the other scenario. Okay? So why do you want to increase rate of respiration? To release / produce more energy. Okay. Now, we will accept both release and produce. Why release? Because the energy is actually trapped in your digested food. Okay? Technically, you can't really produce energy because energy can only be converted from one form to another. It cannot be created. Number 4 point, to meet the increased energy demand of running. So this word running okay, is basically can be replaced with exercise. So it depends on what exercise the question is talking about. It could be swimming, it could be rope jumping, it can be anything. It depends on the question. Okay, then the next part is some of you may go, Mr. Chong, it is very long, it is just 1 mark. I'm telling you that this is a very standard question. It can worth 2 marks. So what are they judging you on? It depends on the question. But to be safe, this is what is required of you to write when the question is talking about, asking you about why there is an increase in heart rate during exercise, the whole thing. Okay, and then you don't miss out on anything. And you realize that it's very relatable. The first point is related to the next point, and then to the next point, and then the next point. Alright? Ken? Okay, next. Alright, moving on to the next question. Okay, this question, we've done it before, and yet and yet it was very, very, very poorly done. So we need to then ask ourselves why. Now, they told you already, Mary prepared two setups as shown in a diagram and left them in a dark room. After a few hours, Mary observed that the lime water in both setups turned chalky, but the lime water in setup A was less chalky compared to B. Now, when you do questions like this, you must always identify your changed variable. So your change variable is basically the condition that got changed, okay, in the experiment. So the difference would be the presence of plants, okay, and the absence of plants, right? Now, then after that, what is your measured variable? Your measured variable will be your lime water, how chalky it is. Now, they told you that carbon dioxide turns lime water chalky. And they told you that, here's the key thing. They didn't say one turn chalky, the other didn't turn chalky. They said both turn chalky. Okay? So they said, explain the changes in color of lime water in both setups. So you have to account for both. So did both setups actually change color? The answer is yes. They both turn chalky. So you have to account. Okay, the first point, how come both turn chalky? So for the first part, you have to tell me, okay, the mice, okay, in both setup A and B respired or carry out respiration. If you cannot spell respired, releasing carbon dioxide, which turn the lime water chalky. Okay, so this explains why both of them turn chalky. Then you have to explain how come one is less chalkier or less chalky than the other. So you have to then explain the second point, because they told you that this is less chalky, right? So the reason, remember the change variable? The hint is probably got to do the change variable, right? There's nothing else, everything else is the same. So it has to got to do with the plant. So think, there is light. They purposely put the light there to hint you. So the light will actually tell you that photosynthesis, okay, would probably occur, right? So, and then you start to think back. When plants photosynthesize, what do they take in? They take in carbon dioxide. So because of that, the amount of carbon dioxide that is here would then decrease because it went into the plant for photosynthesis. Alright, so then you have to articulate that in your answer. So the second part of the answer would be, okay, the plant in set up A took in carbon dioxide for photosynthesis. Hence, reducing the amount of carbon dioxide in the surrounding. This causes the lime water in setup A to be less chalky than setup be? Alright, so as you can see, there's a two-part question. The first part is addressing why the lime water turned chalky in both. The second part of the answer addressed the part as to why A was less chalky, because the plant took in carbon dioxide for photosynthesis. Therefore, the surrounding inside the setup or inside the tank has been reduced. So the lime water will be chalky, less chalkier as compared to the lime water in B. Now then the next question asks you, state one variable that needs to be kept the same for both to ensure fair test. Now this, I'm going to take a little bit of time to unpack this. This is so important. Now there are three things that you need to know about: accuracy, fair test, alright? Okay, accuracy, fair test, okay, these two. And one more thing would be your, one second, let me think. Accuracy, fair test, and sorry. Okay, I'll let you know once I recall that, but the main thing about it is your fair test has got to do with your change variable. Okay, I'm going to write this down. When you talk about fair test, you're looking at the change variable. Okay. Then after that, when you talk about accuracy, okay, that is got to do with your measured variable. And then when you talk about, the last one I remembered, reliability. Sorry, one second, Okay, reliability of results, you're talking about, okay, how many times do you repeat that experiment? Okay, or rather consistency of results. Now, okay, let me explain this to you. This is very important now, super important. Now, the first one, fair test. Fair test is talking about your change variable. That means in an experiment, there can only be one change variable. Everything else must remain the same. So, for example, if you want to find out if drinking milk makes you taller, you will need to have a set of twins. It cannot be two different person of the same age. No. Everything must be exactly the same. Alright? Okay, I'm using something that's very extreme, but I think that's one of the most effective way, because something that you all can think of it every day. So the question would be, will drinking milk make you taller? So you need a set of twins. Everything else about them must be the same. Their CCA, what time they study, what time they sleep, how much sunlight they get, how much air condition. Everything must be the same. The only difference will be one of them actually gets milk and the other does not. That's about it. And that's your change variable. Okay? Measured variable will be your height. Okay? Now, your accurate results has got to do with your measured variable. Yes, your measured variable. Now, what do I mean by that? Let me ask you a question. If I ask you to measure, okay, a road, let's say the corridor, from one end of the corridor all the way to the other end of the corridor, and I give you, okay, a meter ruler, okay, and then ask you to choose between a meter ruler and a measuring tape. Okay, which is more accurate? The measuring tape, right? Because one shot you pull, the way you obtain your measured variable is more accurate. Another example, if I ask you to count the let's say during the Olympic Games, you are trying to find out what is the timing of a swimmer. Would an analog stopwatch, that means those that doesn't have digital screen one, okay, be more accurate, or one that has digital timing that can go up to 2 decimal place be more accurate? 2 decimal place, right? The digital stopwatch. So this will tell you how the instrument that is used or the method of obtaining your measured variable, okay, will affect your accuracy. Now, reliability refers to the number of times you repeat your experiment and you get the same result, consistent. It does not mean that your experiment is fair, neither does it mean that your experiment is flawless. It just means that the results that you get can be repeated by other people. Because that's the whole point, right? Imagine you do an experiment, you found out something. You want to share your findings with the entire world. But first things first, the other part of the world, say I have Professor X from Japan and Professor Z from US. So Professor Z actually found out something amazing and want to share with Professor X in Japan. So he sends Professor X this whole long list of things what to do. Professor X must be able to repeat and get the same results, because then I know it's consistent. Another example will be your vaccine. So they tell you, right, your vaccine needs to be reliable. Why? Reliable in what sense? When you say someone is reliable, so it means that it will keep repeating the results. So yes, so when you get your vaccination for COVID-19, means that it has a certain percent that you can get protection, you will not get the disease. So you want something that's very reliable. Okay? So if I were to be very extreme and tell you this, if your experiment has an error, I can still say that your experiment is reliable, your results are reliable, as long as I can repeat the mistake every single time. Doesn't matter. I'm not dealing with fair test, I'm not dealing with accuracy, I'm dealing with getting the same results. Okay? So the one change variable, okay, kept the same. So the only thing you cannot keep the same is the presence or absence of the plant. However, most of you, right, you will just say things like this. Okay, the lamp. This is wrong. Okay, what you meant to say was the type of lamp. There must be some quantifying word at the front. Or you can say, some of you go, the lime water. No. The quantifying word will be the volume of lime water. Okay? So you cannot just say the container, okay, or the glass cover. You must tell me the type of glass cover, right? Because there's so many. Okay, are you talking about the size? Are you talking about the thickness? Are you talking about the type? You know, there are different kinds of glass. There's stained glass, there's colored glass, there's tempered glass, okay, tinted glass. So the thickness of the glass, they are also different, right? So there must be a quantifying thing. And please do not absolutely in red tell me same location. No. This is a big no. Why? Now, you see, let's just say, what's a location? A lab is a location, a classroom is a location. So if I conduct my experiment on the table in the lab, and then I put it on the second one, I repeated it on the floor of the lab. Are they in the same location? Yes, it is still in the lab, but they're different parts. Then you think about it, the one that's on the table maybe got a bit more fan. Okay, so it's cooler. I don't know, but my point to you is no. Okay? Next. Now, this is also an extremely common question. One look at it, I see this word, water droplets. It signals to me that condensation has happened. And because the water droplets are found on the lid of it, it signals to me that the lid is the cooler surface. Okay? Now, so for this question, right, there isn't exactly a heat source. Okay? But, however, right, there are two answers. I'm going to write down an answer that I think could be a little bit more accurate, okay, and easier for you to understand. Now, you need to understand that inside this whole part here, your surrounding air, that contains water vapor, right? So, one type of answer is to say that the cooler warm water vapor in the surrounding actually gain heat from the steam buns. Right? When it gains heat from the steam bun, the cooler water vapor will become warmer water vapor, rises up, and then it touches the cooler surface of the lid, lose heat, and then condense. That's one way. The second way, okay, would be that you just tell me that water inside the bun, okay, has evaporated to become water vapor. Now, the heat source, there's not really a heat source in the second answer. That's because your the buns has already been taken off the fire, okay, and placed on this plate. So the heat source was your fire, whatever, but you removed it already. So, but is it still warm? Yes. Evaporation still happens. So remember, evaporation happens at any temperature. So because of that, you can just straight away say water in the bun evaporated. Okay? So you can say water in the bun. Of course, if there is a heat source, you must mention the heat source. Must. Okay? Okay, water in the bun evaporates into warmer water vapor. Right? Then the water vapor, okay, rises and comes into contact or touches the cooler surface of the plastic lid. Then we say it what loses heat and condense into water droplets. Now, many of you are still forgetting to write lost heat for some reason. Okay? Next, suggest one method to prevent the water from dripping onto the buns. So some of you said things like, remove the lid. That's not the point. If you remove the lid, guess what? There is no water in the first place, right? So your answer would be to poke holes, okay, into the plastic lid. Okay, that's one way of doing it. I will accept that. Now, when there are holes in the plastic lid, the water vapor can then escape. Right? So when it escapes, less condensation would happen. That's one way. Another way would be actually to put a piece of paper, okay, to just on top of the bun, so that the water droplet would drip on top of the tissue paper. Okay, now you cannot put something that is waterproof. Okay, so your tissue paper is still quite porous. It will still allow the hot water vapor to escape. Okay, but when the water droplets drip on top, you will absorb it. Alright? Next. State one difference between evaporating and boiling. Now I'm going to tell you two. Now the first thing would be, okay, evaporation, get this right, happens at any temperature, not any time. You all tell me any time, any temperature below boiling point. But or while boiling happens only at boiling point. So BP stands for boiling point. Now if you've written 100 degrees, I've given you correct, because at your level you only learn about water. But guess what? Many of your substances, they also boil, but they have their own boiling points. For example, gold boils at a type of a specific boiling point at, I can't remember, a few thousand degrees. Okay, your nitrogen boils at another temperature. Okay, now a second answer would be that boiling occurs throughout the liquid, but evaporation only happens at the surface of the liquid. Please write both of this down. So if you are missing one, write the other one. Okay? I want all annotations to be written down. Okay? Okay, next question. The next one talks about, okay, this person called Ronaldo wanting to dry his hand after washing them. So when you want to dry something, right, immediately that should flag off the concept of evaporation. Yes, so basically your annotation here will tell you. Okay? Now, they said that Ronaldo's hand will dry after a few minutes. Explain how his wet hands became dry. So like I told you, the word dry would have hinted to you that this is a process of evaporation. So in order for evaporation to happen, you need something known as heat, correct? So your hair dryer basically is hot. Okay, so you need to say that, okay, the water on his hands gain heat from the hot air and evaporated. So it's just not enough to tell me that water evaporated. And then after that, if you want to talk about the wind, it's not just enough to say, or rather it's not right to just say, oh, the wind blow away the water droplets. No, you have to tell me that the wind increases rate of evaporation. Okay? Next. Now, then after that, they tell you they conducted an experiment to find out how temperature affects the time taken to dry a pair of wet hands. So obviously at this trend, right, when you take a look at it, okay, as my temperature increases, what happens over here is that my time decreases. Alright, write this down so that you know that it is an inverse relationship. That means when one goes down, the other goes up. So state the relationship between the temperature of air, okay, and the time taken to dry. So because it's an inverse relationship, so you would say that as the temperature of air from the hair dryer or hand dryer increases, the time taken to dry a pair of hands, of wet hands, decreases. Alright, very straightforward. Now for the last question, when Ronaldo increased the wind speed. Okay, so we know that this whole question is on factors affecting rate of evaporation. So wind speed is one of them. Then they ask, will the time taken for his can to be completely dry increase, decrease, or remain the same? Your answer should be decrease. Why? Because you know that wind actually increases rate of evaporation, and evaporation is the key process of drying your hands. Okay? So what's important over here is you need to tell me that it will be to decrease. Some of you, you're not very careful. Okay? You actually say increase because you think of faster. But it's actually not correct. It's decrease, shorten the time. They're talking about time. So be very careful. Decrease. Why? So you have to say, okay, increase wind speeds of the hair dryer or hand dryer will increase the rate of evaporation of the water on his hands. Okay, so obviously when you evaporate at a faster rate, his hand would dry faster. Now for the next question, I'm not going to talk about it because it's actually not being tested, so we will skip that. We have not learned about that. So that's the end of it.